Okay, can you hear me in the back? I assume yes. Wonderful. Okay, my background is not computer science. I'm mainly a biologist. I mean, I got a PhD in biology, and I spent most of my day, talking with and working with neuroscientists. So I noticed that most of you had cake. That's good. That means you're totally awake, but you're probably a little, drowsy in about 5 minutes when the sugar goes down, which is perfect. That's where I'm hitting my first content snag in here. Anyway, so I thought instead of just going through a PowerPoint presentation or something like that, I'll start off with more or less showing you what kind of data we're dealing with, and then I'll explain to you, why we're using Semantic MediaWiki and where we're going really with this. Let's see. Okay. So this is a movie that we're usually showing off, and I'm gonna step here out of the bound of whatever this mark here is on the ground to kind of give you an impression of what we're dealing with. I hope the resolution is somewhat okay. Let's wait and see. So when we're talking about building a network, what we're actually talking about, we're talking about neuroscience data. This is a real cell. This is a cell that was experimentally obtained by somebody in a lab. What they did is they put an electrode here, they recorded how the cell behaves electrically, then they injected a colorant, and somebody went and reconstructed this cell in 3D. So when I'm talking neural networks, I'm talking real neurons, not your artificial neural networks where you got point neurons and they're somehow connected, but this is real biological data. So when we're building our networks, they're really based on a lot of data that gets obtained in a lab. One of it is, the shape of the cell, the morphology. Because if the top of the screen here is the top of the cortex and the bottom is the bottom, depending on where you're sampling cells, you really get cells that have different shapes, different morphologies. And that's important. When people are talking about neuroscience, everybody assumes there's this one type of cell and that's it. In just this small space, 2 millimeters high and about 2 millimeters wide, there are 55 different morphological cell types. That's it. I mean, we're talking the size of a pinhead. That's not the entire brain. That's just a little bit of neocortex. Nothing special there. But as you really look at some of these cells, they look distinct. And the shape of the morphology really gives you a really good feel of, that they are different. And not just that, but they actually have a certain role in this network. If you are a cell like this, you, for example, are able to form connections to the very top. And when you're then back placed into space, what you end up is you start to have intersecting branches with other cells that are in the area around you. So what you see here is what we're doing when we're actually talking about modeling a piece of cortical tissue. What we're doing is we're placing these cells back into the places that they came out of in the appropriate fractions. The red cell is the cell that gets currently placed, and it's, to say profoundly, it's a shitload of wires in there. What you're seeing here is about 10% of what's going on in a normal brain. In a normal brain, you have in 1 millimeter cubed of your brain, you have 4 kilometers of wires. You got that, story from people that talk about the alveoli in your lung. I mean, everybody's high school, bio teacher, if you take all the alveoli in your lung, you could make it to the moon and back. You could wrap the moon a couple of times, still come back and not realize that a part of your brain isn't there anymore. So this is the Indiana Jones view of the model. Again, it's just 10% because if it were 100%, it would be more or less, a solid wall hitting you in the face, and you would be like, wow, cool. So the next thing is really, like I said, you got all of these branches, and that's important because that's where these cells can make connections with each other. And the area that I showed you before, that's a small neocortical column, a microcircuit really. It's composed of 30,000 cells. Okay, great, can do with that. If you multiply that with the number of connections that could exist between a number of these cells, you all of a sudden go from 30,000 cells to 30 million connections. And that's where you then need some of the hardware that we're using to actually model this. You need a supercomputer just to build this kind of thing. And since it's a model, you can pull out any one of these cells and say, okay, great, now I want to see where this cell has connections to other cells. And I want to see these connections in different representations. For example, red is an excitatory connection, that there's a cell somewhere here that tries to pass a signal onto that cell, hoping that it will, pass the message on to somewhere else. If it's a blue bubble, it really just says there's another cell that tells you, oh, look, that's not important. Just forget what the other ones are telling you. Don't say anything. And all of that's kind of cool, but it gets much more fun when you put dynamics on top. This is a multi-compartment model electrically of how a cell behaves. So all of a sudden you see that you have parts of the cell reacting, and you see here the change in membrane surface potential. So what you see is that different parts of the cell can behave differently depending on where they are, depending on who's next to them. And the fun part really starts when you don't just have a single cell, because a single cell is like, yay, wonderful color. But when you have 10 cells, all of a sudden you realize why these connections and why these wires are super important, because that's where signal propagation happens, from one cell to the other cells. And with 10 cells, you can still follow that along. Okay, this one fires, and the next one happens. Okay, great, wonderful. But like I said, we're doing simulations, routinely that have, more than 30,000, 50,000, 100,000 cells. And all of a sudden, you can't follow this thing anymore, but you see interesting things all of a sudden happen. That this wave of activity is something that we didn't put in. The cells start to become, go in synchrony with one another because their time constants are starting to synchronize. That all of a sudden you have certain activity in there that we didn't put in. It's just the way the system is built, it allows you to do certain things. And that's really where one of the challenges of this type of research comes in, that, yeah, great, I can look at everything in there. Now, what's important to look at? You could look at every cell activity in here. You would get, a plot of 50,000 cells doing something. And the important part here is really saying, well, great, but, how do we know that any of that is real? So what you need to do from that is you build a model of what happens when you put all of the data back together. To see if this is anything real, or if this is really an artifact and how we're building it, you need to be able to ask questions that you would normally ask in a laboratory. If I put an electrode here and put an electrode here, do I see the same kind of behavior as I see in the biological system that we're trying to model? Oh, is this thing going to sleep here? Oh, perfect. And that's really where the data-driven modeling approach comes in, of saying, great, well, anytime new data comes in, anytime there's new information about the activity of these cells, about the shapes of these cells, about the connections of them, it's easy. We can just rebuild the model. It's a bunch of parameters that actually allow us to configure these simulations. And the limit on that kind of stuff is really saying, okay, great, I have now what I can build right now. Well, this is actually what we could build a couple of years ago. The rest is really just you add more compute power to it. You can build bigger circuits. You can build more complex regions. And what you see here is really just a demo circuit that if you were to run this kind of complex simulation on the biggest and best machine around, you could simulate the blue part here. This is the neocortex of a mouse. We're talking about, the size of the top part of your, thumb, size-wise. The rest, all the other parts, the lower brain functions, optic nerves, and all these kinds of things, we can't. There isn't a machine around. It's just not possible. But it will be possible in the next couple of years. I don't need to tell you guys anything about compute power doubling and all that kind of stuff. But we will be able to build a simulation at one point that will have the capacity to potentially do, volume-wise and complexity-wise, what we would expect a human brain to be able to do. Now, again, we're not going to do anything crazy here. This thing is not going to talk. It's not going to give us the lottery numbers or anything like that. It's really just saying, okay, well, we want to understand everything that we know about this. Because if we don't put all of the stuff back together, we're screwed. You have then, individual researchers digging in their little corner of the universe, that is neuroscience, characterizing this one cell type, but you never have the wider community actually profiting from that knowledge. You don't have anybody profiting from that data, because the only thing that comes out of all that research is a paper. 10 pages summarizing a year of research, and that's it. And when that person retires, the janitors of the university clear out the cabinets and throw all the stuff out. And that's the reality of academic research a lot of times, that a lot of that knowledge leaves with the person that leaves. Now, what we've done, and we meaning the BlueBrain, which is a part of the Human Brain project, Well, I mean, to clarify, the Human Brain Project is just a large grant. It's not a group, it's not a structure, it's not anything, it's a grant. A grant that since October 1st is coordinating 86 partner universities in Europe and a couple of partners in the US and in Asia to try to achieve that, to say, okay, look, what would happen if we put everything together that we know right now about, the neuronal system in the mouse, in the rat, and what happens if we pull everything together that we know about human data. And again, don't freak out here. If I look at the number of publications around that explain to us what we know about rat, mouse, and human, we know very little about human actually, because there aren't a lot of experiments performed on humans. And for some of the data, you will not get human data. You don't have too many volunteers saying, yeah, just open up part of my brain, pull out a cell. So the important thing is really we need to first know what we know, and that is about everything in the mouse, in the rat, and in the human, and then say, look, if we were to do a simulation like this on human data, what are the holes? What are the things that we're missing? And is there equivalent data in the mouse or in the rat that would give us an idea on what this might look like in human? It could just be that the morphologies of the cells are similar in human and in mouse, but the size is different, the numbers are different. A number of these things are known, a number of them are assumed. What we're trying to do is we're trying to get everybody together to say, look, let's start pulling all this information together. And again, the problem is that a lot of that information is out there in papers, but the papers are really just an abstraction of the data that's around. So to give you an idea on what we're trying to do, we started to collect a lot of information over experiments that have been done in the last 2 decades in the laboratory that really started to fuel these first draft simulations of what I showed you here. All of it is based on rat experiments, and most of it is cell morphological data. And we're trying to move that from very dedicated databases to something more generic, something where anybody in the world that has this kind of data about, mouse, rat, or whatever other organism sees that, well, they can share this information if they choose so, and that there's actually very little overhead for them to start using some of the tools that we have lying around for taking a look at the data, analyzing these things, because a lot of neuroscientists are not computer scientists. They're happy, with Excel, they're happy with Word, but a number of these tools are not really up to the task of, complete analysis of everything that we know of a certain data type. One example of that is if we say, okay, well, let's look at all the cells that we have available right now in a database, and I hope this loads. It's one of the dangers of a live demo. We're all staring at the screen, but only I'm sweating. Okay, this is embarrassingly slow. Hang on, let me see if that does anything. It might help.
Try another hotspot.
Okay. Ooh. Let's switch back here. I swear it was working 15 minutes ago. Anyway, while we're waiting for that to not do anything, maybe I'll go into a little bit of detail of what we're using Semantic Wiki for. The problem really is that when you start to capture multiple aspects of data, that you actually have a distributed process, that you have one person that works on the shape of the cell, another one works on the location of the cell, the third one actually works on the reconstruction of the cell, and that we found it actually makes a lot of sense if everybody works on their process, but that you use the structure of the Semantic MediaWiki, that once all of that information is available, you have an easy way of pulling it all back together, of saying, well, great, you have this type of information of the morphology of a cell, you have this location of the cell, you have the electrical behavior of that cell, and all of them live in their own little data structure. But a lot of times I actually want to get an overview of what looks like when I want to see everything that is known about this cell, about this one experiment. That I can say I would like to see the overview, or I would like to dig deeper into a detail. And this is not going good.
Perhaps there is somebody with 3G that can do USB tethering.
Or does anybody have Wi-Fi switched on and Chrome installed? I'll do the demo on your laptop.
You try to refresh the page.
Well, I still don't have Wi-Fi.
Maybe you have to put in the login.
Can you get this possible?
Sure.
There you go. Okay. How does he? There's no transcription on this? Or can I get the track test out?
Okay, there we go. So, for example, if I want to take a look at all the cells that I have right now available in the database or in the wiki, really I can just say, okay, well, I would like to go to the level of, cell from here. So I have organisms, I have microcircuits, I have micronetworks, I have cells, I have subcellular. Those are really just different levels, depending on where I fix my microscope. And I can look at all the cells that I have available right now. Oh, sorry. In here, and those of you that know about these dynamic galleries will recognize that these are really just images with properties on them, and that's it. So what I can do is relatively easy. I upload the images, they get the proper tags, and that's it. And what I get immediately is I get something that is more intuitive for an experimenter to look at. Instead of trying to memorize ID numbers, they can just look for their favorite cell and say, okay, great, whoop, there we go, this is my cell. I want to know more about this cell. I actually want to see if everything that I put in there got captured. And what I see is, hey, you only tell me it's a pyramidal cell. What about the location? Where's my location? Why didn't you put that there? Well, it's not there. You didn't put it in the system yet. It actually allows us to get a very quick global overview where the individual researchers can say, well, okay, great, this doesn't disappear into some kind of, black hole database that they don't know how to edit, they don't know how to augment, they don't know what to do with, but that this is something that is very accessible to them and where they can just, say edit. They go in, they change things and say, look, my comment is something happened here, the person that reconstructed the cell did a bad job, they did a good job. And to show you an example of what actually happens when you have a relatively complete record, what you could get is you could get something like this of saying, well, this was a cell that was captured as part of an experiment or of a project. And being a complete naive user, not knowing what this thing is, I can just say, okay, well, let's go explore. I want to see more about that project. I want to get some kind of, summary. What did they do there? And that, yeah, here I get my abstract that actually describes to me what they've done in this project. And a lot of projects, for example, could be 2 weeks, 6 weeks, 8 weeks. It could be a year, it could be 5 years. I want to know where it was done, what laboratory was it done in. I would like to know who are the people that were involved and what aspect were they involved. Because if I don't find any information on, for example, the morphological classification of that, I want to know who I need to talk to. Also, I want to see links to the raw data, because like Yolanda said, sometimes the data is just too big. Sometimes you cannot put it in the wiki. Sometimes you're talking about gigabytes, terabytes of imaging information that you wouldn't want to look at in the wiki. You would like to see that this exists and features that were extracted out of that, but sometimes you're just as fine just looking at the analysis. You might have a modeling component involved in this of saying, well, we can explain this data if you actually were to model the information that came out. This is what would come out, an abstract model. And the curation aspect, okay, you have a lot of information there. Is all that, did that go through the proper process of curation? Are we happy with this? Is this something that is still in process? And again, different levels, organisms, brain regions, cells, molecules. And what we found is that it's incredibly flexible, incredibly dynamic, because again, I can go back to my cell, which is where I started, and say, okay, great, I want to know more about this one cell. Well, here's my summary. I can find out that it got classified based on 3 different systems. And if I want to know more about it, I can say, okay, go, show me the location. Show me actually what we know. What's the basis of the location that was assigned to that cell? Well, it's an image, and somebody identified the cell up here. That's my basis for what the information is that I have in here. And it's very transparent for the user of saying, look, this is not something magical that we're doing in the back with the keyboards. If you want to look at the morphology of the cell, yep, there you go. Sorry, we're still playing with viewers and this kind of stuff. That the process that you can see is, well, this is something that somebody reconstructed in the lab. They went and reconstructed the cell. Based on this, you can then have a process run that extracts features out. And that can happen every time that somebody contributes that piece of data to the wiki. Then you run this process of getting the features out. Based on that features, a second process gets then triggered that classifies the cell. And in case you want to go back and take a look at this with a different tool, there you go, here's a link, download it again, analyze it, improve the process, and we'll re-trigger it, we rerun it. There's no problem. And just because we like people in the lab, this thing is dynamic. There you go, move it around, do whatever you want with it, and you don't even need the program that people use to build this. It's easy, drop it in and you immediately get benefits. You get this across multiple different areas of saying, okay, look, electrical activity of that cell. There you go. Spike, 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 spike. Wonderful. You can do the same thing, for example, for connectivity. Okay, well, we see that there's a connection between those 2 cells because there's a squiggly arrow going there. That's what we know about that cell. And again, just to go with some of the wiki aspects in here, okay, great. This cell was actually part of a connection. So this cell is a presynaptic cell. It's the cell that gives the signal to another cell. And here's the postsynaptic cell. And if I want to know more about that, okay, there you go. I just click on it. It actually gets people to look at a lot more things because it's become increasingly easy. Sorry, again, the viewer. You can say, okay, great. Now, yeah, come on. There you go. That's a second cell in that connection. And all of a sudden you got people going like, hey, that's kind of cool. So what happens if I put in another cell? What if I actually want to look at 3 cells, 5 cells, 12 cells at the same time? Yeah, you can. It's as easy as saying, well, look, there is no view that exists for this right now, but how would you like to see your data? Would you like to see, for example, all the networks at the same time that were ever recorded? There you go. Again, just going to another overview page of saying, how would you like to navigate this information? Would you like it, with a visual overview? Would you like to look at it as a list? How can we help you make sense of the data that you put in and compare it to what we're doing in the end? That you might want to go from, say, a protocol and say, okay, well, I have a certain process that I'm implementing in a lab. I would like to see data that was produced with this process. That you just go in and say, okay, great. Now show me the process. Process number 1, wear a lab coat and put on latex gloves. I mean, things that people in a lab would actually do, not some kind of abstract paragraph long an experiment was performed. The solutions that were used in this, the equipment that was used, the procedure itself, and then really saying, okay, look, give me an example of what data looks like that comes out of this process. Where somebody that actually performed the experiment can go back in and say, this is not as good as I thought. Maybe I don't want to do this process. Maybe I want to use another process to get this type of data, because what comes out here is not what I expected. And the same thing for analysis protocols, that you really can say, look, there are people involved in this whole workflow that have varying skill levels. We got somebody that actually, there you go, that came up with how to do this classification of cells, because that's what they do. They're not machine learners, they're not statisticians, but that's what they did when they, open up the electrical traces and said, look, first what I do is I look at the slope of this. If the slope is greater than that, does this, does that. Where this can then go back to somebody on our more computational side and say, look, I can write you a classifier for that. We can do this automatically. You don't need to do this by hand anymore. And the only thing that changes is that when you throw in now your data record, this runs automatically. So you get immediately a benefit out of showing us what you're doing, showing us how you're doing it, and asking, look, can you do something for us? Can you make our life a little bit easier? Because we spend hours and hours doing this by hand. If you had a classifier that actually does what we want to do, we'd be grateful, and we'd be able to contribute much, much more to this, because you have a loop going between people that have data, people that have analysis, and people that then do something with this data in the end. Because this cycle that we're doing is a lot bigger than a lot of normal curation happening in wikis. Everything that comes out of this wiki is the input to simulation. So depending on how good this data is here, the simulation will be better or will be worse. If you don't have this cycle going back and forth, you actually run into the problem that you might model a system that doesn't exist in its current form, because the people that provided the data said, look, here is what we did back then, but we're not going to go in and reclassify because this is just too hard for us. We don't understand how this process works. And that's what we're using Semantic MediaWiki for, that we actually make a lot of these processes more transparent, of saying, look, this is what happens with the data. This is the first step. You put it in. Next step that happens is something computational. You get features out, you do a classification. Does that agree with how you think things are working? So we can actually work more fractured, but all work on the same topic at the same time, really. Okay. I think time's up. Sorry. Yes.
So is that not publicly readable, and if not, why?
It's not publicly readable because we ran really into the problem that the beginning of the month we showed this to everybody else that is part of the Human Brain Consortium, and we immediately had about 2,000 people that wanted to use it right now, immediately, for their data. And that we're 70 people working on that. We're ready to do development, but we're not quite ready to do community support for people that are burdened with Excel. So it's one of those things where we're saying, okay, we'll make it available, we make it accessible, and all the data that's coming out of the Human Brain Project will be publicly accessible. All the analysis, all of that will be there.
Okay, great.
Mm-hmm. Yep.
Terrific presentation. I'd like to know about your performance relative to how much data you're putting in and the size of the hardware, this sort of thing.
Right now, we're not running into a lot of problems with that yet. It's kind of funny because we're try to do this with a plain vanilla Semantic MediaWiki. So there are no extra extensions. There's nothing on top, and we really just try to figure out, look, does it make more sense having sub-objects versus pages? Because, for example, some of the things that we have in here, we're pushing a lot of limits there with saying, okay, well, if I load in every recording of a single cell, for example, and that cell gets captured by molecular process, I might have 25,000 links per cell. If you do that for 42 cells, you have 1 million links, and you can have either 1 million pages or 1 million sub-objects. And that's where we're kind of trying to figure out, what is the proper way to do this? Should we create pages? Should we do sub-objects? And that's why I'm here also, because I want to get a feel of where the community is going with this kind of thing of saying, well, look, great, but just move that into, you know, a database and, you know, go with a template on top of that.
I think the kind of data you have is a very good example of the kind of data where it, well, it's worth considering whether a Wikibase would be a better approach than Semantic MediaWiki, simply because this kind of data usually, you often have sources. You want to say where that data came from,
right.
On a very fine-grained level, right? For individual properties, you want to be able to qualify something, saying something was derived using such a method, for instance. Even for numbers you have in there, you may want to say what the accuracy of that number is, and so on. And all of these things are built into Wikibase.
The part that I didn't get to really was that on top of this whole thing, a part of the numbers that go in and the data goes in, one thing that we have is we've tried to use Wikipedia and a normal media wiki for getting neuroscientists to start, you know, characterizing their cells, characterizing their brain regions. It doesn't work. The problem is that when they go to it, they see the page and they go like, great, now I got to start from scratch. Why do the work in this one? Because if I do it in a normal paper, I get credit for publishing. If I just put it on Wikipedia, I don't get any credit. The hook that we have is that we split the screen. And I'm sorry, I'll try to bring this up here. It's the sorry, nope. Sorry if I'm exposing links here that I'm not supposed to show, Yuri. If I go to, sorry, microcircuits. This is really the part that, I mean, that's one of the parts that keeps people actually engaged of saying, look, I have my living review here. I have what we think we know about this thing right now. And on this side, I got data streaming in of saying, great, this is what you got out of a paper. And this is actually what the data tells you. That this can be a parallel iterative process. Saying, well, great, well, from the data we have, for example, a mean height, a standard deviation, a unit, and a number. You don't have that in the review. Okay. There you go. So as an expert, you don't even need to look a lot of times for the data. You can just go and say, great, I have this coming in. And that this is something that I haven't seen yet, where you say, look, you can have parts of the information really come from a data wiki, from a database. And parts of it is this community-driven refinement of what we know, qualifying also the information on that side. It could be that you're looking at this and saying, look, that's really a bad value. And I think it's because the method was bad that they used. It's not the number itself that was bad, but they fucked up somewhere here. How can I qualify that? How can I bring that back into this process of saying, maybe the number is wrong?